Good evening, everybody. Yesterday we did the part 1 of immunology, and today we are in the last part, that is the part 2 of immunology. I told you yesterday that this is antibody, and these antibodies, they are 5 types. Firstly, I told you there are 3 fragments: FV, that is heterodimer; FAB, it is also heterodimer because it consists of, that is, heavy chain and light chain, whereas it is homodimer, it has only the heavy chain, and it's a hinge which separates the FAB and FC. And these are basically the antigen binding sites. And FC, I already told you that it is the area where basically when the person is allergic, having allergic reactions, and it is the mast cells and basophils which are formed in response to allergic, they are attached to this FC region. These are the, I'm reviewing and giving you the abstract. So how the questions will come. So I already told you there are 5 types of heavy chain. On that there are 5 types of antibodies, that is alpha heavy chain means IgA, and that it has 15% in blood serum, and this is antibody found in mother milk secretions like tear, mucus and saliva. This question will come. And the first defense against invading bacteria and virus, it is dimer, whereas the IgD, it is monomer, where IgE is also monomer, and IgG, they are 3 monomers. IgE, I just told you it is formed due to the allergic reactions, whereas IgG, which accounts for the maximum, this question will come, 50 to 80% of total antibodies present in serum, and IgG can pass the placenta and provide passive immunity. This question will come, whereas IgD does not pass through the placenta, and it helps the baby to initiate the immune response. So one antibody, that is IgM, is the pentamer, and it is also of maximum size, that is 900 kD, and 5 to 10% of total antibodies, and when exposed to antigens, IgM molecules appear first. That indicates disease which is caused by the pathogens. And yesterday, then other thing I told you, there are basically 2 types of cells, right? And these 2 types of cells we did yesterday are the first is the that is cytotoxic T cells, and we have the helper T cells. So in that we have particularly is the formation of Th1 and Th2, and Th1 and Th2 which is formed, and Th1 produce I already told you the interleukins or cytokines, which is IL-2, and the most important is IFN alpha, whereas the IL-10 is the now discovered that Th1 and Th2 both produced, and this results in the B cells forming the IgG, and but in Th2 the IL-4 it changes the B cell path from IgG to IgE. So this question will come. And then we have IL-5 here, which results in the formation of eosinophils, or I already told you which are the produced in during the allergic reaction, and they are attached to the FC region. So this we did, and no issue with that. And then I told you then the MHCs I told you there are 2 classes. First is class 1, that consists of single polymorphic transmembrane. This question will come, where MSC2 there are 2 polymorphic membranes, whereas in case of it is made up of 2, that is alpha, it is 44 kD, and extracellular, that is beta 2 which is coded outside MHC, and alpha is divided into alpha 1, 2, 3, and it is the 3 where the CD8 binding site is there. whereas the MHC2, which have two polymorphic peptides, is alpha and beta. This is 12 kD and this is 31 kD. It divided into alpha 1 and alpha 2 and beta 1 and beta 2. Right? It is the beta 2 where the CD4 is attached. So in case of MHC2, CD4, whereas in case of MHC1, CD1. And after that, I told you yesterday also, so it is the CD4 also function as a receptor for HIV, that is, which is main culprit of many deaths, that is AIDS. And this helps permitting the virus to enter the helper T cells, and then it paralyzing the immune system, right? And then MHC molecule present foreign antigens to the cytotoxic T cells, which kills the infected target cells having class 1 MHC. So cytotoxic T cell only identify the MHC1, right? So it is the Nobel Prize winner like Rolf Zinkernagel and Peter Botany in 1986, right? So next type we did is the cytotoxic recognition type. That is, first is the viral RNA comes. It is endocytosis in the membrane, and then it attached to the endosome, and here the fusion of virus when endosome membrane and escape of viral into the cytosol. Very important, because a few minutes I will ask you one question. So replication of viral RNA and then proteolysis of viral RNA, that is proteasomes, which are coined by the MSC genes, and it is changed into the peptides, and they move to the basically transport to the ER lumen, endoplasmic, where it combined with the beta microglobulin and MHC1, and then they transfer to the Golgi, where it comes to on the surface, and it is recognized by the cytotoxic T cells, right? Cytotoxic T cells, it recognizes MHC1, and it is basically the recognition by the cytotoxic T cell is facilitated by protein called talin. This question will also come, right? So now today I will do the second part of it, that is the recognition of the that is by helper T cells. The topic is the helper T cells and T cells activation. So it is same like that we did yesterday, but here we have the folded protein antigen. There we have viral RNA. Here is folded protein antigen. It undergoes plasma membrane endocytosis, and it is taken by the early endosome. Early endosome, it is a simple structure like this, right? Where late endosome, it is basically wavy and it is rough and not smooth, and it is irregular in shape. And here in this helper T cells, basically through their receptors are recognized by the class 2 MHC associated with antigens and displayed on the surface of target cells, right? So see, then we have in this Golgi apparatus. So basically the class 2 MHC molecules are also synthesized in endoplasmic reticulum, just like class 1 we did yesterday, right? See, this is class 1 in MHC, that is ER, MHC1 in endoplasmic reticulum. So it is also synthesized in endoplasmic reticulum, right? So the class 2 MHC molecule is actually pre-assembled and through trans-Golgi network reaches the late endosome. So it basically this is the trans-Golgi apparatus, and through this it reaches the late endosome, right? And basically these when it reaches the late endosome, where it associates with the peptide fragments, right? Same as that of earlier we did. See here endoplasmic, it is these are the peptide fragments, right? So it basically then associate with the peptide fragments using its binding groove, right? So the binding groove is initially occupied by a dummy peptide. So that binding groove where these MHC2 they attach, they are before that they are basically occupied by a dummy peptide called clip. This question will come, called clip, right? So this clip, which is derived from the invariant chain, this one, right? Thus preventing the ER, means endoplasmic peptides, to bind. So that groove to be occupied by the clip, so that no endoplasmic reticulum peptide can take that place. And in the endosome, clip is released by proteolysis. So proteolysis, that same thing we did here. Proteolysis results in the release of that clip. We did in yesterday also. Here, see, this is proteolysis. So here with proteolysis, the clip is released, and now groove is accessible to the peptides fragments. And these peptide fragments, they are derived from the, as we already told you, endocytosed proteins, right? So these peptides, they combine with the, then as clip is released, and they will combine with the MHC2, and then this moves towards the plasma membrane, and it comes out, and this is recognized by the helper T cells only when it is accompanied with the MHC2. So it is obligatory for the recognition, the presence of MHCs, which we did yesterday. That is the major histocompatibility complex, that is class 1 and class 2, right? So these are its types. And again, that is alpha 3 where CD8 is attached binding site, and beta 2 where CD4 is attached, right? So helper T cell and activation, the helper T cells do not kill the infected target cells as done in cytotoxic T cells, but instead either stimulate macrophages to destroy pathogen. So helper T cells, like we did yesterday, they do not kill the infected target cell, they stimulate the macrophages, right? So I already told you. So it is two type of cells we have, that is the B cells. B cells, they results the humoral immunity, whereas T cells, without any mediation of antibodies, they form cellular immunity, and T cells, they migrate to and mature in thymus, whereas the B cells, they matures in the bone marrow. Yeah, this we did yesterday. So now we are in continuation with that class 2 MHC molecules are also synthesized in ER just like MHC1 molecules, but the difference is the formation of a that is occupation of that groove with the by means of a clip, right? So thus class 1 molecules presents peptides originated in cytosol. This second difference is that class 1 molecules, they present peptides originating in cytosol, that we did yesterday. See, the viral DNA, endocytosis, endosome, and from endosome fusion they come to the cytosol. I told you I will ask you one question. Where MHC molecules, they present peptides that comes from the extracellular space through endocytosis. See, these are coming from the extracellular space through endocytosis, right? These are the two differences we have. And then we come to the abstract part, that is very important. I just did in the 3 categories, and I already revise you that yesterday I told you Seidberg coefficient. So all the antibodies, they have 6 to 11 except the IgM, which has 19S. Here 3 factors proteins. First is CD3, and expressed on target cells is all T cells, and ligand is no ligand here, whereas CD4, it is helper. Just we did CD4, and I told you the attachment of point of CD4 is the MHC2, that is C, MHC2, and that is beta 2, right? Then CD4, we have helper T cells, and that is class 2 MHC, that is beta 2, and that beta 2 is binding site for CD4, whereas CD8, that is cytotoxic T cell, class 1 MHC, and CD8 is attached to the alpha 3, right? So this I already told you, this is the alpha 3, it is the CD8 binding site. So now I am coming to CD28. CD28 is basically in many helper and cytotoxic cells, that is the B7. This is very important. So it is bound to the plasma membrane, and C28 has a ligand B7, which is bounded to the transmembrane, and it's expressed on the many helper and cytotoxic T cells. So now I was just winding it up. That is the immunoglobin I already told you. So immunoglobin are basically having the three fragments, that is the FC and FAB. They are both heterodimer and contain both that is heavy chain and light chain. And whereas the FC, which is the separated from FB by a hinge region, is homodimer because it consists only of heavy chain, that is CH3 and CH2 domains. So these FV and FAB, they are basically the point where the antigen will strike. So next I already told you, then IL-10 is the present in both Th1 and Th2, whereas IFN, that is interferon gamma, is the most prominent factor, which is cytokines. I already told you in COVID era, when people are suffering and they don't recover and the body said it is enough, it's excretes cytokines. And these cytokines not only kills the pathogen, means coronavirus, but also kills the organs. That is, people are dying due to the multiple organ failures. So Th1, it has IL-2, which has CD8 T cell, and IFN gamma, which produce macrophages, whereas Th2, which produces IL-4, which change the B cell formation to Ig1G to IgE, right, which is important for the allergic reactions. Then IL-10 is present in both in Th1 and Th2. This question will come, and IL-5 will result in formation of eosinophil. So exogenous or extracellular, it is MHC class 2. Endogenous and intracellular, it is abstract MHC class 1. Cross protection, in which the exogenous antigens can be presented by MHC class 1. Okay? And this way finishes the immune system, and any problem you will face, you can revert back. So next I will start the biotechnology topic, right? So introduction of biotechnology, and then the medias. So I just want to blend between the biotechnology and molecular biology, because these biotechnology and molecular biology are the topics which plays important role in whether you are clearing the UGC or not, right? So 90% questions, they comes from the biotechnology and molecular biology. That is why I am blending it down. And I assure you, if you see these all videos, it is become easy for you to clear. And then I just circulated book also, the book for the reference. So besides that, any problem you will face, you can revert back to me, and we will solve that problem together, right? And the main aim of our college, that is me and our worthy principal, Professor Dr. Anita Kaushal, is to get you out of the competitive exams. We want you people will clear maximum of competitive exams, so that it will be easy to for you people to foot into the word of the competitions, right? So now I request you, please make our hand stronger, just to read through that all that I am just teaching you, and be ready for the fourth series of mock online test, that is blend of UG and PG, that is basically on the July 26, right? So stay indoors, stay safe, take care, and please revise these things, right? And any you will feel any problem with this dealing, just revert back to me, right? Take care, stay indoors, bye bye.